Welcome back to the deep dive. We take complex biology, break it down, and give you that concentrated knowledge you need. Today, it's a critical mission. We've got sources here detailing, well, the essential targets when a cell gets injured. And there's one thing, one structure, that really decides, the point of no return. We are diving deep into cellular membrane damage, that moment when the cell's ultimate border just breaks. Think of the cell membrane as not just skin. It's like a super smart security wall, a shipping center, and it organizes the power grid, all in one tiny package. If that fails, the whole thing collapses. That's basically irreversible injury, leads straight to necrosis. Okay, let's unpack this. Our sources, they paint this picture of total system failure. It's not just like one hole. It's like a perfect storm hitting the outside wall, the power plant, and the garbage disposal all at once. We are looking at what happens when the cell's security system totally breaks down.
Yeah, that holistic view is really key. The main thing across all these sources is that the first most consistent sign of lethal injury, well, except for apoptosis, it's different, is losing selective permeability. The cell just can't control what comes in and what goes out anymore. And like you said, the damage isn't just in one spot. We need to... The membrane damage isn't only the outer plasma membrane failing, it's also those crucial internal barriers, the mitochondrial membranes making the power, and the lysosomal membranes holding all those potent digestive enzymes. It's how these failures are all connected, the mechanisms causing them. That's what we're really digging into today.
Right. So the cell's a fortress, basically, with 3 critical walls that can be breached. We should look at each one, understand what it does, and what happens when it fails, catastrophically. Hey, let's start with the plasma membrane, the outer wall. This keeps the environment stable, right? So its failure means instant osmotic chaos. Why does losing energy, say, from lack of oxygen or a toxin, hit the plasma membrane's integrity so fast?
Well, it really boils down to active transport. Specifically, the sodium-potassium pump, the TEXTA plus ATPase. This thing uses a ton of energy, constantly working. It's one of those ATP-hungry parts of the cell. It keeps sodium low inside, potassium high inside. That steep gradient is vital. So the second ATP production tanks, maybe during ischemia or, a big dose of toxin, that sodium pump just stops, dead in its tracks, and that failure. It's immediately catastrophic. The gradient starts to collapse instantly. Sodium ions, which are packed outside the cell, they just flood in, down their concentration gradient. And water just follows the sodium osmotically, rushes right in after it.
So the cell just floods, fills up like a water balloon.
Exactly. That leads directly to that visible cell swelling, what doctors call hydropic change. And internal stuff swells too, like the endoplasmic reticulum. It dilates. It's a rapid internal flood, a huge water imbalance that stretches the cell, sometimes past its breaking point.
But it's not just unwanted stuff getting in. The sources really stress this dual disaster. Vital things leak out. They specifically call out glycolytic intermediates leaking out. Why is losing those particular molecules such a tipping point? Why is that irreversible?
That's the final nail in the coffin for energy production. See, when the cell's ischemic, it desperately switches to anaerobic glycolysis. It's not efficient, not at all, but it's an emergency power source. It doesn't need oxygen. But to do that, it needs those intermediates, the building blocks for that whole pathway. So if the plasma membrane is leaky and these vital intermediates escape, well, the cell loses even its emergency backup fuel. It can't make any meaningful ATP, not even anaerobically. Losing that last shot at making energy just accelerates the whole crisis. Makes recovery pretty much impossible. The damage becomes, well, irreversible.
Okay, moving inside the cell now. B, mitochondrial membrane damage, the power plant barrier. If the plasma membrane's the outer wall, the mitochondria are like the inner sanctum, where the real energy gets made. Why do the sources call mitochondria the arbiters of life and death?
Because they are incredibly sensitive targets. They react to almost everything: injurious hypoxia, definitely ROS exposure, massive calcium overload. They're where the cell's fate is ultimately sealed. The key physical failure inside the mitochondria is the opening of something called the mitochondrial permeability transition pore, the MPTP. This thing is a huge channel, high conductance. It doesn't need to stay open long to cause total disaster.
And once that pore opens, boom.
Yeah.
2 different ways the cell can die, right? Decides if it dies and how. Let's start with the messy one, necrosis.
Right, that path is paved to just running out of energy. When the MPTP opens, the electrochemical gradient across the inner mitochondrial membrane collapses, Immediately. That gradient, the proton motive force, that's the engine-driven oxidative phosphorylation, the process that makes like 95% of the cell's ATP. So gradient collapses, oxidative phosphorylation just stops cold. The cell runs into a massive terminal ATP shortage. All the functions that need ATP fail rapidly, uncontrollably, and that inevitably leads to necrosis, that messy, explosive kind of cell death.
But the same kind of injury can sometimes make the cell choose a different path, a controlled demolition apoptosis. What's the subtle difference in that mitochondrial failure that triggers the intrinsic apoptotic pathway instead?
Yeah, the difference often comes down to the type of damage and, importantly, what specific proteins get released. In apoptosis, the damage makes specific mitochondrial proteins leak out into the cytoplasm, things like cytochrome C. Once that's out in the cytoplasm, it kicks off the caspase cascade. That's the cell's own execution machinery. This leakage, it's often helped along by pro-apoptotic proteins like BAX and BAK. They actually form channels in the outer mitochondrial membrane, letting things out. So yeah, both involve mitochondrial damage. But necrosis is driven by that total ATP collapse after the MPTP opens wide, apoptosis. That's driven more by the leakage of specific kill-me signal proteins. It's the molecular context of the failure that nudges the cell towards one fate or the other.
Okay, that brings us to wall number 3, the one containing the self-destruct button. C. Lysosomal membrane injury, the suicide bag. If the cells somehow survive the flood through the plasma membrane and the power plant failure in the mitochondria, why is breaching the lysosome the absolute point of no return?
Because lysosomes contain the ultimate demolition crew, a huge arsenal of really potent hydrolytic enzymes. They're designed specifically to digest complex molecules. We're talking about acid hydrolases. The list includes RNases, DNases, proteases, phosphatases, glucosidases.
Wow, that sounds like a toolkit designed specifically to dismantle a cell, piece by piece.
That's exactly what they do. So when the lysosomal membrane breaks, these enzymes spill out into the cytoplasm. And here's where all these terrible mechanisms link up beautifully, or horrifyingly. The cell, remember, is already failing energy-wise. It's shifted to anaerobic metabolism. That produces lactic acid. So the inside of the cell gets more acidic. The pH drops. Now, this acidic environment, which is bad for normal cell functions, it's the perfect condition for those leaked lysosomal enzymes. The low pH activates them. They immediately start chewing up everything essential: RNA, proteins, DNA, and the nucleus. Once that self-digestion kicks off, it's terminal. The cell is pushed irreversibly into necrosis. It digests itself from the inside out.
Right. Okay, we've mapped the 3 disaster sites. Now let's switch gears to the how. Here's where it gets really interesting. What are the actual biochemical weapons? The mechanisms coordinating these attacks often hitting all at once. Our sources point to 4 really interconnected mechanisms that basically guarantee membrane failure. A. Reactive Oxygen Species, ROS, and Lipid Peroxidation. We just start here. ROS, these oxygen-free radicals. They're central to so many injuries, especially things like ischemia-reperfusion. What exactly is a free radical? Why is that unpaired electron so dangerous for a lipid membrane? Okay, simply put, a free radical is just a molecule with a single unpaired electron in its outer shell. Electrons desperately want to be paired up, so this makes the radical incredibly unstable, highly reactive. It will aggressively steal an electron from any stable molecule nearby and the cell membrane. It's packed full of nice, stable lipid molecules, perfect targets. Now, normally during respiration, the cell makes some ROS, but it has systems to neutralize them quickly. The problem comes when those systems fail or get overwhelmed, like when oxygen suddenly comes back after ischemia, that's reperfusion injury. You get this burst of radicals, especially the hydroxyl radical, nasty stuff. That leads to oxidative stress.
And the main way they destroy the membrane is this process called lipid peroxidation. How does that actually work? What's the mechanism? Right, lipid peroxidation. It's an autocatalytic chain reaction. Think of it like rust, but happening incredibly fast and spreading. The hydroxyl radical loves to attack the double bonds in unsaturated fatty acids. Those are common in membrane phospholipids. When it attacks, it steals an electron, oxidizing the lipid. This creates a new unstable lipid radical. Now, this new lipid radical reacts with oxygen, forming a peroxide radical, and that peroxide radical then attacks the lipid next door, stealing its electron. And on it goes.
Like a fire spreading through the membrane.
Exactly like that. The initial attack is the spark. But the peroxides formed, they act like more fuel, more sparks. The damage propagates, spreads like wildfire through the membrane bilayer. It causes massive, irreversible structural damage, makes the membrane leaky. It's not a localized thing, it's a systemic breakdown of the membrane structure.
Okay, that's chemical attack. The second mechanism ties right back to that energy crisis we talked about.
Yeah.
B, decreased phospholipid synthesis. This is the cell basically being unable to repair itself under fire.
Yeah, it's a difference between routine maintenance and emergency repairs during a siege. You need a constant supply of materials to patch the walls if they're being constantly attacked, right? Well, making phospholipids, which are needed to build and repair all three crucial membranes, plasma, mitochondrial, lysosomal, that whole process is incredibly energy hungry. It needs a lot of ATP. You need ATP for almost every step, activating the raw materials, moving molecules around, driving the enzymes. So when mitochondrial function tanks or hypoxia cuts off oxygen, and ATP production just plummets, this energy-dependent synthesis pathway is one of the first things to get shut down.
So even if the cell is only taking minor hits from ROS, if it doesn't have the ATP, it can't even patch the small holes. It's like a passive failure compounding the act of destruction.
Precisely. The cell loses its ability to do basic upkeep and repair. The total amount of phospholipids in the cell drops. This makes every membrane structure weaker, thinner, much more vulnerable to all the other chemical and physical attacks happening at the same time.
Okay, now the third mechanism, C, increased phospholipid breakdown. This sounds more aggressive. And this is where calcium really steps up as a major culprit, right? This isn't passive failure. This is active demolition of existing membrane.
Oh, absolutely. This is probably the clearest link between calcium going haywire and direct damage. Like we said, severe injury ischemia, toxins leads to this massive uncontrolled flood of calcium into the cytosol, text 2+2. This calcium surge switches on a specific group of enzymes, the calcium-dependent phospholipases. These phospholipases literally start dismantling the membrane phospholipids, chewing them up. But the fallout is twofold. First, obviously, you lose the structural lipids holding the membrane together. Second, and this is just as bad, the breakdown process itself creates toxic byproducts, lipid breakdown products.
Toxic byproducts, what are they and how are they toxic?
The sources name things like unesterified free fatty acids, acylcarnitine, and lysophospholipids, and they describe these molecules as having a potent detergent effect on membranes.
Detergent effect, you mean like soap dissolving grease? The cell membrane.
Exactly like that. Remember, the membrane's a lipid bilayer. Detergents work because they can insert into that lipid structure, messing up the hydrophobic and hydrophilic interactions, essentially dissolving it. These toxic lipid breakdown products. They do the same thing. They wedge themselves into the remaining lipid bilayer or swap places with intact lipids. This drastically messes with permeability, fluidity, electrical properties. They act like molecular crowbars, basically, prying the membrane apart from the inside, dissolving its structural integrity.
And finally, the fourth mechanism. D, cytoskeletal abnormalities. This sounds more like a physical failure. Damage not to the wall itself, but to the internal steel beams, maybe.
Yeah, that's a good analogy. It speaks to the cell's overall physical strength. The cytoskeleton proteins like actin, spectrin, they act as internal anchors. They connect the fluid plasma membrane to the more stable structures inside the cell. They're crucial for resisting physical stress, especially the stretching force from that osmotic swelling we talked about earlier.
All right, so the cell is already swelling up because the sodium pump failed. Those anchor points are already under strain. Now, bring calcium back into the picture.
Right, that same massive calcium influx that turned on the phospholipases. It also activates another set of enzymes, calcium-dependent proteases, called calpines. These proteases specifically target and break down the protein scaffolds, the cytoskeletal anchors. So now you've got this triple threat hitting the plasma membrane. It's swelling up from water influx, hydropic change. It's being chemically degraded by phospholipases, and now its internal support structure is being actively dissolved by proteases. This combination leads to a catastrophic physical failure. The plasma membrane literally detaches from the underlying cytoskeleton. The outer wall, unsupported, stretched thin by swelling, chemically weakened, becomes incredibly prone to just tearing, rupturing. In cells that experience a lot of mechanical force, like heart muscle cells, this physical rupture is often the final visible blow that signals necrosis. So, if we take a step back, look at how all these mechanisms fit together. What's really fascinating here is how these four really different kinds of attack, chemical assault, energy starvation, enzymatic digestion, structural collapse, they all funnel down into just two core problems. A profound failure of energy production, so ATP depletion, and massive uncontrolled calcium dysregulation.
Let's zero in on calcium, the master switch for destruction. We mentioned that calcium in the cytosol, free text Ka2 plus Broer, is normally kept incredibly low, like 0.111 micromolar. Why is keeping it that low so absolutely vital?
Because calcium is arguably the most important second messenger inside the cell. It triggers muscle contraction, drives signaling pathways, controls secretion. It's essential for life, but only in tiny, tightly controlled bursts. If that tight regulation breaks down, calcium turns from essential messenger into a universal poison. Ischemia, toxins, physical injury, they all cause way too much calcium to build up in the cytosol. Some flows in from outside, sure, but a lot of it is released from internal stores, especially the endoplasmic reticulum and, critically, the mitochondria.
And once it hits those high levels, it doesn't just do one bad thing. It kicks off a whole destructive feedback loop, doesn't it?
Oh, absolutely. It simultaneously activates pretty much every major destructive enzyme we've talked about. It hits the phospholipases, breaking down membranes. It hits the proteases, damaging the cytoskeleton, causing detachment. It even activates endonucleases, which start chopping up the DNA in the nucleus. And here's the kicker. It also activates ATPases, enzymes that just burn through ATP. So not only is the calcium actively causing destruction, it's also rapidly consuming whatever little precious energy the cell might have left. This guarantees that vicious cycle. Calcium causes damage and speeds up the energy failure that prevents any hope of repair. It really is the final common pathway that seals the deal for cell death by necrosis.
Okay, let's switch from the attack weapons to the defense systems.
Yep.
If making ROS is just a normal part of breathing, the cell must have an incredibly good defense to maintain oxidative balance. To stop oxidative stress, that state where free radicals just overwhelm everything.
Exactly. The cell is constantly battling ROS. So it's evolved these really sophisticated ROS defense systems. They're multi-layered. They're strategically placed right where the oxidants are likely to be generated. We can think of these defenses as having two main arms: specialized enzymatic scavengers and more general chemical antioxidants.
Let's start with the enzymes. The first line of defense may be the most critical: superoxide dismutases, SODs. What's their specific job?
SODs are absolutely essential. They're metalloenzymes. They need metal ions like manganese or copper and zinc to work. Their job is to tackle the superoxide anion, TXO2. That's the most common free radical made during normal oxidative phosphorylation in the mitochondria. SOD takes this really reactive superoxide and converts it into hydrogen peroxide, XO2, which is much less reactive. And notice the location. There's manganese SOD right inside the mitochondria where most ROS are made. And then there's copper zinc SOD out in the cytoplasm. This placement is key. Neutralize the threat at the source before it spreads and starts that lipid peroxidation chain reaction.
Okay, so SOD makes hydrogen peroxide, dextrose toler, but that still sounds potentially bad. Isn't hydrogen peroxide involved in making the really nasty hydroxyl radical, tovo-HN, through the Fenton reaction? How does the cell deal with the TXO2?
Good point. That's where the next enzyme comes in, catalase. Catalase is mainly found in little organelles called peroxisomes. Its job is basically dedicated TXO2 disposal. It works incredibly fast and efficiently. It takes hydrogen peroxide and breaks it down into completely harmless water, TXO2, and oxygen TXO2. So catalase makes sure the byproduct of SOD's work doesn't become a new problem.
Right. And the third major enzyme defense is glutathione peroxidase. It sounds like it overlaps with catalase, but maybe does more.
That's right. Glutathione peroxidase is also critically important. It can reduce hydrogen peroxide to water like catalase, but it can also tackle more complex lipid peroxides, those damaged lipids that have already formed in the membrane. So it's not just preventing damage, it's helping clean up damage that's already occurred. It does this using a molecule called reduced glutathione, or GSH. GSH gets oxidized in the process. In fact, the cell's balance of reduced glutathione, GSH, versus oxidized glutathione, GSSG, is a really important indicator. Clinicians sometimes measure it. If the balance shifts towards the oxidized form, it tells you the cell is under heavy oxidative stress. Its defenses are struggling.
Okay, beyond these powerhouse enzymes, the cell also uses simpler, small molecule chemical defenses. The non-enzymatic scavengers, or antioxidants. These are the things we hear about in nutrition all the time, right? Vitamins and such.
Exactly. And they're crucial because they work together with the enzymes. It's a team effort. These include the fat-soluble vitamins, like vitamin E and vitamin A. They actually sit within the lipid bilayer of the membrane, physically positioned to intercept radicals and break that lipid peroxidation chain reaction right where it starts. Then you have water-soluble scavengers out in the cytosol, things like ascorbic acid, vitamin C, and glutathione itself. They neutralize radicals floating around in the watery parts of the cell. It's this really complex, multi-layered defense system operating in the cytoplasm, inside mitochondria, within the membranes themselves, that has to stay fully functional just to keep the membranes intact during normal life, let alone under stress.
It's really clear the sources focus heavily on how membranes break, defining necrosis by this structural and chemical collapse. But the flip side, and this is a huge area in modern cell biology, is how complex membrane repair is. The cell actively trying to fix the damage before it's too late.
Yeah, that shift in focus is vital.
Yeah.
Because that's the difference between reversible and irreversible injury, right? A cell can often survive a short, small hit like brief ischemia. But that survival depends entirely on its ability to quickly patch things up, to restore membrane integrity. That often means plugging small holes, replacing damaged phospholipids.
And if you loop back to our source material, that ability to repair hinges on two absolute must-haves: having enough leftover ATP and keeping that calcium level under tight control.
Absolutely. The repair processes themselves need energy. For instance, patching a small tear often involves something called TXK A2 plus triggered vesicle fusion. When a tiny hole occurs, a little bit of calcium leaks in locally. That small calcium signal helps mobilize internal vesicles, little membrane sacs, to move to the plasma membrane and fuse with it, basically slapping a patch over the hole. But moving vesicles, fusing membranes, making new phospholipids for the patch, all that takes energy. Needs ATP. So if the initial injury drops ATP levels below what's needed for repair, and at the same time causes that massive system-wide calcium flood that activates all the destructive enzymes, well, the cell's trapped. It's stuck in a downward spiral. Damage happens faster than repair can keep up.
Which brings us back to that key difference between necrosis to the explosive mess from membrane failure and apoptosis, that controlled self-destruction. The membrane failure really dictates the outcome.
It really does. Think about inherited diseases involving DNA damage sensors. Cells have systems, like the p53 pathway. They sense if the DNA is catastrophically damaged, maybe by radiation or free radicals that got into the nucleus. When something like p53 gets triggered, the cell often makes a programmed decision to undergo apoptosis, usually through that mitochondrial pathway we discussed. The cell basically says, look, living with this damaged DNA is too risky, could lead to cancer, better to commit suicide cleanly. And during apoptosis, the membranes mostly stay intact. No big leaks, no inflammation.
But the kind of massive membrane damage from overwhelming ROS and calcium,
Yeah.
there's no choice there.
No choice at all. It's just violent, uncontrolled rupture. The sheer extent of the membrane damage hitting the plasma membrane, the mitochondria, the lysosomes, that's what fundamentally separates the controlled dismantling of apoptosis from the messy, inflammatory explosion of necrosis, the integrity of those walls. It's the ultimate decider of whether the cell lives or dies and how. And, if we connect this to the bigger picture, the battle for the membrane is truly the battle for the cell's survival. That synchronized failure, ATP vanishing, calcium flooding, and those are the final common pathways. They destroy the internal structure, breach the outer wall, leading directly to what we actually see under the microscope as necrosis, the massive swelling, the organelles disintegrating, everything leaking out.
Wow. This has been a really deep dive, a fantastic journey right to the edge of cellular life and death. We've clearly established that membrane damage isn't just one thing failing. It's this complex, multi-pronged attack hitting the plasma, mitochondrial, and lysosomal membranes. And the key takeaways seem pretty clear. Once that selective permeability is gone, it's probably lights out. The destruction comes from this unholy alliance, ROS causing runaway lipid peroxidation, ATP depletion, stopping any synthesis or repair, and that massive calcium overload turning on every destructive enzyme imaginable. It's just this rapid, irreversible spiral. Energy crisis, physical damage, chemical breakdown. We really hope this deep dive into the cell's broken border gave you those aha moments and new appreciation for just how critical those boundaries are.
And maybe just to leave you with a final thought, something to chew on, drawing directly from how essential these defenses are. The sources show just how vital membrane integrity is for life itself. So consider this. If making ROS is just a normal, unavoidable side effect of making energy every second, and yet ROS are also the key weapon in causing rapid cell injury, just how incredibly finely tuned do those natural antioxidant systems like SOD and catalase have to be just to cope with normal everyday breathing at the cellular level? And then what happens to those membrane defenses when the systems start to decline, maybe due to things we can't easily control, like just getting older or certain chronic diseases? That balance, that's maybe a deep dive for another time.